Topoisomerase, helicase, DNA polymerase, and ligase all play a role in replication, which is a semi-conservative process. So this idea that it's semi-conservative, what that means is that we start with our original strand, and the two new strands each are made up of new strands. So we get a little bit of the old strand in the two new strands. So again, as we see this, we've unzipped the original DNA, but then off of each half of the original, we get this newly synthesized daughter strands that are building, so it is semi-conservative. So as we go here, all the different players involved. First of all, we'll start with topoisomerase. Now again, this is going to relax the supercoils in front of what we call the replication fork. And it's at this fork, this split, where we have helicase. And helicase is going to basically unzip the DNA. We also have primase, which is a form of RNA polymerase, and it has to lay down an RNA primer before DNA polymerase can start adding newly synthesized DNA. DNA polymerase cannot build from scratch. It can only attach the new DNA nucleotides to existing nucleotides on the 3' end. So RNA polymerase, or this primase, has to first lay down an RNA primer so that DNA polymerase has something to grab onto. Ligase, and again, what it's going to do is join these fragments together on the lagging strand. So on the leading strand, DNA polymerase is building continuously into the fork. However, on the lagging strand, DNA polymerase has to continually loop back into the fork and build away, because DNA polymerase can only build in one direction. So then ligase attaches all these fragments together and helps with the covalent bonding of those backbones. So if we think about bacteria and prokaryotes and eukaryotes, this would be a prokaryote. Its DNA is a circular ring. And when replication begins, it has two replication forks going in opposite directions until ultimately it finishes that process. And again, this is still semi-conservative, and you have replication complete, and it stays in its ring structure, circular chromosome. Now with eukaryotes, we end up with these replication bubbles. And again, eukaryote DNA is linear, so you end up with these replication bubbles that in multiple places that have multiple replication forks, and they ultimately grow and they connect until we have the new daughter strands formed. So one more way to look at this, again, a couple other terminology. When we say the DNA strand is anti-parallel, what that means is that it goes from 5 to 3' in one direction, but then in order to have base pairing work, it has to be 5 to 3' in the other direction. So that leads us to having to, because it is anti-parallel, we end up with having to have leading and lagging strands. But first of all, the 3', again, when we think about a nucleotide, we have these carbons in the sugar. And on this end of the sugar, you have a hydroxyl end. And that's what we call our 3' end, where the hydroxyl group is. And the 5' end is where the phosphate group attaches. So we have 3' and 5'. Now, again, if we're going through this whole process, topoisomerase would have to first relax the supercoiling. And then we would have helicase unzip, and it would be, this is where we would consider our replication fork. Now, the original strands are read in the 3 to 5' direction, which then that puts DNA polymerase here and here, because the 3' end is right by the replication fork on the lagging strand, and it's away from the replication fork on the leading strand. Now again, I'm leaving primase out of this, but it would first have to lay down that RNA primer before DNA polymerase could grab on. But again, this is our leading strand, and this is our lagging strand. This is going to be leading because DNA polymerase is going to build into the replication fork on this one, and DNA polymerase is going to build away from the replication fork and have to re-loop. So when the newly synthesized strand is being built, it has to build in the 5 to 3' direction because DNA polymerase can only add onto the 3' end. So this is, the original is 3 to 5 here, but again, this new strand, this is the 5' end right here, and DNA polymerase is going to add on to the hydroxyl group on the 3' end. So again, it's building 5 to 3. So this DNA polymerase here would be the 5' end, and it's going to build onto the 3' end where the hydroxyl group is. So it would continue adding nucleotides at a time, and if it ever made a mistake, it could back up and fix it and then rebuild. So DNA polymerase is able to correct itself and double check for errors. But DNA polymerase would continue on, adding on to 3' ends until it has that strand complete. Now again, this process would continue until it's done, so we'd continue unzipping it, and on the leading strand, DNA polymerase would just build into the replication fork. But it's at this point where DNA polymerase would have to re-loop back around and catch up to where it was unwound and build back towards this end. And it would be at this end, after it catches back up to where it started, that ligase would connect those Okazaki fragments. So again, this process is what we call semi-conservative because we have the original strands that are being conserved, but with each new DNA template, we end up with a new daughter strand built onto the original strand. So helicase, topoisomerase, DNA polymerase, and ligase all play a role in replication, which is a semi-conservative process.